Okay, welcome to the video for chapter 7.5. So 7.5 is all about photosynthesis and carbon fixation. So we're going to talk about the differences between photosynthesis in microbes versus photosynthesis in eukaryotic cells like plants. Okay, but first, let's do some review questions. Okay, review question number 1. E. coli is a human pathogen that can use many different types of metabolism depending on the environment. List the types of metabolism in order of most energy, so ATP produced, to least. So is it A, fermentation, aerobic respiration, anaerobic respiration, B, anaerobic respiration, fermentation, aerobic respiration, C, aerobic respiration, anaerobic respiration, fermentation, or D, fermentation, anaerobic respiration to aerobic respiration. Okay, so pause the video, look at your notes for 7.4, and see if you can't find the answer. Okay, so before I give you the answer, let's look at these responses. Okay, so we know that the first one has to be the most energy produced. Okay, so which one of these processes makes the most energy? Well, we know that that's aerobic respiration. So aerobic respiration has to be listed first. So if we look at our options, our first option here is fermentation. Automatically, no, we know that one's the least. So A is out. Okay, B is anaerobic respiration. That's not aerobic. B is out. C has aerobic respiration, so this is a contender for the right answer. And D starts with fermentation. Well, if A is not right, D is not right. So the only possible answer this one could be is C, aerobic respiration. This is a good question to know for the exam, and it can be easily flipped, right? So if this question said that to order the types of metabolism in order from least energy produced to most, well, then we're going to need fermentation. So A or D is going to be the answer. But we know that anaerobic's the middle guy. He makes more than fermentation, but less than aerobic. So the answer for that type of question would be D. So really good question to know. Okay, so let's go to review question number 2. Review question number 2 says, oxidative phosphorylation requires an electron transport system, or ETS, to develop a proton motor force, PMF, which in eukaryotes occurs in the mitochondrial inner membrane. Bacteria do not have mitochondria. How can they generate an ETC? Okay, so we need to find the answer to this. So A says the ETC takes place in the plasma membrane of prokaryotes, and a proton motor force is generated between the plasma membrane and the cell wall. B, prokaryotes are unable to do oxidative phosphorylation because they don't have mitochondria. And C, electron transport and ATP synthesis takes place in the cytoplasm of prokaryotes. And D, prokaryotes do not need an ETC. They can get ATP from the environment. Okay, so take a second to pause the video and see if you can't find the answer. Okay, so where do we find the electron transport chain, the ETS, for prokaryotes? Well, we find it in the plasma membrane, the cell membrane. Okay, so we need to find the answer that reflects that. Okay, well, the only one that's correct here is A. Okay, it says the ETC takes place in the plasma membrane, check, and the proton motor force is generated between the plasma membrane and the cell wall, in some, yes. But yeah, B is wrong, because we know that prokaryotes can undergo oxidative phosphorylation if they are aerobic or anaerobic. Okay, it's true that they don't have mitochondria. C, the electron transport and ATP synthesis takes place in the cytoplasm. This is no, because we have to have those electron transport chain enzymes embedded in a membrane. And we know D is not correct, because we cannot get ATP from the environment directly. We have to make it. Okay, so the answer is A for this question. Okay, so chapter 7.5 is all about photosynthesis, aka carbon fixation. So how we can fix carbon to make sugars. Okay, so photosynthesis is the way photosynthetic individuals can fix carbon. Okay, so you need to be able to describe how photosynthesis harvests light to generate ATP and NADPH while releasing oxygen. You need to be able to describe how a photosynthetic microbe fixes carbon dioxide into biomass. You need to be able to explain the importance of photosynthesis for your human health. So let's get started. Okay, so let's talk about photosynthesis. Photosynthesis is probably one of the most memorized things in high school biology, no matter what biology class you take or who you take it from. Okay, so we've all learned the photosynthesis equation. 6CO2 plus 6H2O plus light energy is going to go make C6H12O6 plus 6O2. Okay, we all memorized it. We all talked about it, right? If you took biology from me, I made you memorize it. Now, what we don't tell you in biology in high school is that we have different types of photosynthesis. So the photosynthesis equation that we learned in high school biology is what we call oxygenic photosynthesis. Okay, so this is photosynthesis that produces oxygen as a byproduct as well as our organic sugar molecule. Okay, so with photosynthesis, it does carbon fixation. Carbon fixation is where we're going to take an inorganic carbon source, so in this case it's carbon dioxide, CO2, and then we're going to make an organic molecule like glucose from it. Okay, so one of the happy accidents about photosynthesis is that for oxygenic photosynthesis, where we use water as a reactant, we're going to get oxygen gas back. Now, anoxygenic photosynthesis, so photosynthesis that does not make oxygen, is going to make another type of gas. So instead of using H2O as a reactant, we're going to use something like H2S, and we'll make like sulfur gas from it. Okay. Now, one of the things that we need to talk about is the fact that oxygen is a byproduct. It's a waste product for most organisms. Okay, most of the oxygen that we breathe and depend on has come from photosynthesis done by microbes. Okay, so when we're talking about photosynthesis, we're talking about photolysis, so the lysis of something made from light. So we're using sorry guys, we're using light to break something. Okay, so in this case, we're using light energy to split water into individual oxygen atoms. Okay, so we're going to split or lyse water to produce oxygen gas, some electrons, and some protons. Okay, so we have 4 high energy electrons that come from this reaction. Now, all plants and photosynthetic organisms that go through oxygenic photosynthesis are going to do this. Okay, but the thing is, we talked about in chapter 6 how for a lot of organisms, oxygen is very toxic because it's very reactive. So plants and other photosynthetic organisms have this process that once they make that waste product, once they make oxygen, the first thing they do is release it or excrete it as fast as possible so that oxygen doesn't cause damage to their cells. Okay, so this process of excreting this waste product, excreting oxygen, has changed our whole planet. Okay, so early on in the evolution of life, there was very little oxygen in our atmosphere. In fact, we were mostly nitrogen with very little oxygen. Now, today, we're still mostly nitrogen, but we are an oxygen-rich atmosphere because of this process of early photosynthetic bacteria releasing this waste product oxygen into the air. Okay, they literally changed our whole atmosphere that support a lot of aerobic life because of it. Okay, so let's talk about photolysis in a little bit more detail. Okay, so let's talk about carbon fixation, photolysis. So carbon fixation is the process of reducing and incorporating an inorganic carbon into a carbon skeleton or backbone for a biological molecule. So in the case of photosynthesis, our carbon fixation is going to take inorganic carbon dioxide and it's going to make it the backbone of our glucose molecule. Okay, so photolysis is the use of light to break something. Okay, so light is going to be used to generate high energy electrons and ATP, which then can be used to make sugars. So we have this process of photolysis that happens in chloroplasts for plant cells or in different areas for bacteria, which we'll talk about on the next slide. Okay, so what's going to happen is we're going to have our chlorophyll. It's going to capture light energy. That light energy is going to split water. So we're going to get oxygen gas as our waste product. And we're going to get 2 high energy electrons from each water molecule. Okay, so the process of photolysis is going to use an electron transport system and two things called photosystems. Okay, so our electrons are going to go into these photosystems, okay, which are then going to take our low energy electrons and charge them up with light to make them high energy. Okay, so once that's done, and we have these high energy electrons, they're going to go through this electron transport system, which, spoiler alert, just like with respiration, is going to pump hydrogen ions out of out of the membrane. Okay, so we're going to make another proton motive force, which is going to be important here in a second. Okay, so this electron's going to go through this electron transport chain until it gets to the next photosystem. We're going to capture some more light. We're going to recharge that electron, get it high energy, and then it's going to go through another electron transport system where we're going to take NADP plus, which is like NAD. It's a oxidized form of NADP, and we're going to reduce it, so we're going to gain electrons, so we're going to get an electron carrier. Okay, we're going to then take our proton motive force that we have built because of this electron transport system, and those hydrogens are going to then go through ATP synthase, and this is going to take ADP and phosphate and make ATP again, just like in respiration. But the difference between photolysis and respiration is where that ATP goes and where the NADPH goes. Okay, so we have a specific enzyme called rubisco that's going to remove carbon dioxide from the atmosphere in order for us to fix it in the next step of photosynthesis. Okay, so let's talk about photosynthesis in general, and then we'll talk about it in cyanobacteria, and then we'll talk about what we learned in biology, and that's how plants and things that have chloroplasts do photosynthesis. Okay, so in all photosynthetic organisms, whether they be eukaryotic or prokaryote are going to have these thylakoids, which means they're going to have thylakoid membranes that contain chlorophyll. Okay, so chlorophyll is going to be a pigment that absorbs light. Okay, so they're going to absorb light in these grana pockets. Light is going to be used to split the water. We're going to form oxygen. We're going to convert that light energy through that electron transport chain and those photosystems to make ATP from ADP, but also to make NADPH from NADP. There's a typo here. There should be NADP plus. Okay, so this first part of photosynthesis is what we sometimes refer to as the light-dependent reaction or the light reactions, because we have to have light in order for this process to happen. Okay, so we produce energy, we produce ATP and NADPH, so we have our electron carrier, and those are going to go to the next step of photosynthesis, which is what we call the Calvin cycle or the light independent reactions. So we don't have to have light for this piece. Okay, so what's going to happen is we're going to use this energy and the energy in NADPH, and we're going to take our inorganic carbon, our CO2, and we're going to reduce carbon dioxide to fix carbon. Okay, so every time this happens, we fix one CO2 molecule. So glucose has 6 carbons, so this cycle is going to turn 6 times and use 6 CO2s to make one C6H12O6. Okay, so once we have that carbon fixed or reduced, we can then use it in a bunch of organic materials such as sugars, amino acids, and other forms of biomass. Okay, so that's what I want you to start thinking about this product as. This sugar, this organic carbon, is what is going to go to make biomass in cells and different organisms. Now, when we talked about photosynthesis in biology, we talked about chloroplasts. Okay, spoiler alert. Cyanobacteria do not have chloroplasts. In fact, chloroplasts are about the same size as cyanobacteria. They can't have them. So if cyanobacteria don't have chloroplasts, where does photosynthesis happen? Well, the thing is, in bacteria cells that do photosynthesis, what's going to happen is they're going to have folded parts of their plasma membrane that are going to contain these thylakoid membranes. Okay, so these folded region of the membrane are what we refer to as the thylakoids. Okay, so that's going to be where this chlorophyll is contained, and this is where we're going to capture light, is in these folds in the membrane. Okay, so this is a diagram to show you the difference between most bacteria cells and cyanobacteria. So we've talked about this version here on the left a lot, but let's look at how cyanobacteria are different. Okay, so they still have the nucleoid, they still have ribosomes, they have these, let's see if I can say it today, phycobilosomes. So sometimes they're called cyanosomes. So this is where the chlorophyll is found. Okay, so these are the thylakoid membranes, these long structures. Okay, so that's going to look like these folds in the actual bacteria. So this is what the thylakoids, or these photosynthetic parts of these cyanobacteria, look like underneath the electron microscope. Okay, so very different from how plant cells do photosynthesis. Okay, so let's switch gears, because the process of photosynthesis is the same whether you are a cyanobacteria or a plant cell, but in a plant cell we just have chloroplasts. We have a structure that does this a lot like the mitochondria does cellular respiration in eukaryotic cells. Okay, so we have our chloroplast. It is going to be a triple membrane. Okay, we're going to have the inner membrane, the outer membrane, and what we call the thylakoid membrane. Okay, so the inner membrane is going to have an aqueous fluid called stroma, and it's going to contain these interconnected fluid-filled membrane sacs, which we're going to call the thylakoids. Okay, so in cyanobacteria, these are part of the folds in the plasma membrane. We're going to have individual membranes bound in a membrane. So, right, membrane-bound organelles. Okay, so these are our thylakoids. So when we have a stack of thylakoids like this, it kind of looks like a pancake stack, this is what we call a granum. Okay, so multiple granum are called grana. Okay, the thylakoid membrane is what is going to contain the chlorophyll that's going to capture this light energy. Okay, so just like in bacteria, we have the same type of reactions. The only difference is where they occur. Okay, so different parts of photosynthesis are going to happen in different parts of the chloroplast. So we still have the light reactions, so that's going to happen in the thylakoid membranes because we have to be able to catch light. Remember, light reactions are the light-dependent reactions. Okay, so the thylakoids are going to catch light. We're going to get our ATP and NADPH, just like with the cyanobacteria. We're going to produce oxygen from splitting water with light. Okay, that ATP and NADPH is then going to move to the stroma, so that liquid that fills the chloroplast, and that's where the Calvin cycle is going to happen. Okay, so CO2 is going to come in. We're going to use ATP and that electron carrier to fix it, to make it into an organic molecule, and then we're going to produce our sugar, our glucose molecule. Okay, so I would know where the dark reactions happen in the chloroplast and where the light reactions happen in the chloroplast. So where's the Calvin cycle? Now, one of the things to remember is that the Calvin cycle is not part of cellular respiration. It only occurs in photosynthesis. So the TCA cycle and the Calvin cycle get mixed up a lot. So be very careful about that on the next exam. Now, I can say, as somebody who did it when I was first learning about photosynthesis, it's really easy to do. TCA cycle, Calvin cycle, they both use glucose, but one makes it and one actually catabolizes it, right? Harvests all the energy that we can get from it. Okay, so that is photosynthesis. Okay, so let's wrap up real quick what we've learned. Okay, so in biology, you probably memorize a lot more about photosynthesis. We don't need to do that for this class. You just need to know what's in this lecture. Okay, so you need to know that photosynthesis occurs in two stages, the light reactions and the dark reactions, or aka the Calvin cycle. So it doesn't matter if you are cyanobacteria or you're a plant cell, you're going to have the same types of reactions. The only difference between the two is where they happen. So in photosynthetic prokaryotes, it's going to be in those folds in the membrane, and then in plant cells, they're going to be happening in the chloroplasts. Okay, so in stage 1, we're going to capture light energy. We are going to split water to get high energy electrons. We're going to use them to make ATP using ATP synthase, and we're going to make the electron carrier NADPH. Okay, and it's in the light reaction where we make oxygen. You need to know that. Okay, so stage 2, we're going to bring in carbon dioxide from the atmosphere, and we're going to go through the Calvin cycle. So we're going to use that ATP generated in the light reaction and that NADPH, and we're going to pull in 6 molecules of carbon dioxide, and that's going to go to make glucose. Okay, so we're going to regenerate our ADP and our NADP plus, and that's going to go back to the thylakoids for the light reactions to start the process all over again. Okay, so when we make sugar, we're making biomass. So that's a good thing to remember. Okay, so that's really all we need to know about photosynthesis. Let's talk about what happens when we have dangerous photosynthetic organisms. Okay, so let's talk about a case history. Our case history is called Food Poisoned by Red Tide. So this is the effect that some photosynthetic organisms can have on people health-wise. Okay, so let's set the scene. It is June, it's nice and warm, and we're at a beach in Massachusetts. Okay, so we're going to follow Tanya, our 32-year-old tourist who's visiting this town for the first time. Okay, so one of Tanya's bucket list things is when she's visiting this town, she wants to go do clamming. So clamming is the process of literally digging up clams for dinner. So you take your pail and you take your shovel and you walk the beach and you carefully look for little spouts of water to come out of the sand. Because where those spouts of water are, that's where you're going to find your clams. Okay, so as Tanya's digging up her clams and she's clamming, she looks at the water, because as you do at the beach, and she noticed that near the shoreline, the water is clear, but the further away from the shore she was looking, that the water took on this reddish tint. Okay, not a good sign, by the way. Okay, so Tanya finally got a bucket full of clams. She took them home. She cooked them. She's really excited. Bucket list item checked off. But there's a problem. She ate her clams, and shortly after dinner, her lips started to grow numb, and her fingers started tingling. Okay, and pretty soon after that, she felt extremely nauseated, and she started to have difficulty standing up and difficulty walking across the room. She started to get really scared when her speech started to slur, but before things got too bad, she was able to dial 911 and get some help. Okay, so Tanya was put into an ambulance and was rushed to the hospital. Okay, when they got Tanya there, they immediately started to pump her stomach and give her assistance breathing. Okay, she didn't have to go on a respirator, but she got one of the oxygen cannulas to help. Okay, so because Tanya's a tourist, the ER doctor took a minute to explain what had happened. Okay, so Tanya had contracted paralytic shellfish poisoning because she had consumed clams that were contaminated with an algal toxin. Okay, so this toxin is called saxitoxin, and it accumulates in clams as they feed on the algae. Okay, the thing is, with this toxin, cooking it does not affect it. It does not denature it as a protein. It survives cooking. So even though she cooked the clams, she still got exposed to the toxin. Okay, what Tanya didn't know at the time, because she was a tourist, is that part of Massachusetts had been on a shellfish ban because of this saxitoxin producing algae. Okay, it was really high density, so when she was looking out at the water and she was seeing that reddish tint, this is that algae that we refer to as red tide. So the actual algae is called Alexandrium tamarense. Okay, so we're just going to call it red tide. It's a lot easier to say. Okay, so saxitoxin is a neurotoxin that is going to cause paralysis. Okay, and it does this by shutting down the sodium channels in your brain and also in bird neurons. So birds can be affected by this toxin as well because they go clamming as well. Okay, so Tanya's poisoning was all because of this neurotoxin. Okay, so saxitoxin is produced by what we call a dinoflagellate algae. So these are photoheterotrophs, Okay, and these are what are going to make up this red tide. So when you go clamming or go to eat shellfish and there's red tide in the area, you have to make sure that the shellfish was imported from somewhere else because of the danger of red tide. Okay, so that is chapter 7.5. We've talked about photosynthesis. We've talked about its electron transport system. We've talked about how we use light to split water to make ATP and NADPH, and then how we use NADPH and ATP to fix carbon dioxide to make biomass, so to make sugar. Okay, so that's what you need to know for photosynthesis, and we'll see you in the video for chapter 7.6.